Hi everyone. Thank you for the invitation. So I'm Theo from Paris-Saclay University in France, and today I will talk about photobleaching of fluorescent proteins. And it doesn't work, obviously. Okay, it's okay. As we already saw on the morning, fluorescent proteins have this kind of structure with chromophore inside of a barrel. And this chromophore is formed by an autocatalytical reaction that occur during the natural maturation of the protein by the cyclization of 3 amino acids. Why are these proteins so interesting? It's because they are genetically encoded, which means that you can build a DNA that will express both a protein of interest and a fluorescent protein directly inside the cell. So for example, here we have a cell which expresses a cyan fluorescent protein fused to a membrana protein, and then we see the membranes here. So these tools are really important for probe biological events in living cells. So since we discovered the first fluorescent proteins in the medulla Aequorea victoria, we found a lot of mutations to tune the properties of these fluorescent proteins. So the most obvious is to change one of the 3 amino acids that are involved in the chromophore formation to have different p-conjugated systems, so different colors, different optical properties. But the role peptidic chain is important because it can tune a lot of other properties that are also important. And among these properties, there is one which is still difficult to optimize because we don't really understand what's happened, and it's the photostability. Because fluorescent proteins are really likely to undergo irreversible photochemical reactions that we don't really understand, and that lead to photobleaching, aka loss of fluorescence intensity. And it's easy to understand that, except in some very specific experiments, we don't want to bleach our fluorescent proteins when we do fluorescent imaging, because we see here, for example, that we lose very quickly our signal. So we need to improve photostability of existing fluorescent proteins. And that's why my PhD project is to better understand the photochemical reactions that are involved in photobleaching, to pinpoint some residues that are particularly involved in the reactions and which we could mutate to do more, to make more photostable mutants. So for today, I will just talk about a little part of this project, which is just a spectroscopic overview of what's happened when we bleach a large panel of fluorescent proteins. So I will focus on 6 fluorescent proteins. So we have ones that are really close to each other in terms of peptidic chain. For example, here we have only one mutation between these 2 proteins. But we have also different barrel, different colors. We have a lot of diversity on this panel to have the best overview that we can. So that's the pitch. I have these proteins purified in solutions, and now I will put light on it and see what's happened. To do that, I have a dedicated setup which have been made during my PhD project, and which is called BEAM. So it's really cheaper and less sophisticated than a lot of setups that we saw during these 3 days, because it's just basically light which we focus on the sample in the cuvette, powerful enough to trigger the photochemical reaction, and then we will record absorption and emission spectra over the time. Because it's homemade, we have the control on a lot of parameters, which is very important for us. And what's is most important is that we have both spectroscopic and kinetic information, because for each time point, we record the raw spectra. And then, because I work in vitro, I can take the bleached solution, the radiated solutions, and do all the analytical chemistry that I want on it to understand what photoproducts have been formed. So as I said, I will just focus on the spectroscopic part. So this is a typical data set that I can have just in the beam experiment. So first, if we look at the emission spectra, we see that it decreases over the time. So cool, we did photobleaching on all the fluorescent proteins that we wanted to study. That's a starting point. If we look at the absorption spectra, we have more information. First, we see that the absorption main band decreases also over the time, which means that we are not doing some quenching or something like that. We are doing true photochemical reactions, as we expected. Another point is that here we see that for some of fluorescent proteins, most of them, we are forming new species that absorb at shorter wavelengths. And for green and yellow fluorescent proteins, we can identify this species as a protonated form of the chromophore, because it's a tyrosine-based chromophore. It's a phenol. It's fluorescent in the anionic state. It's not in a protonated state. So we know here that we are forming a species which is protonated and new to be non-fluorescent. So it's a part of the explanation of what is the photobleaching here. But we also know that all that we lose here is not converted here because we know the absorption coefficient relative of this species. So if we try to begin to classify what we saw, we have in the bleached solution the remaining FPs that have a new absorption band, which I will call colored, and some species that doesn't absorb anymore because we see that it's not enough to explain. But we have also other things that happen, because when we look now at the kinetics, we see that for all fluorescent proteins, the fluorescence decrease faster than the absorption, which means that what we described here is not enough to explain the whole photobleaching. So we have species that absorb normally but does not emit fluorescence anymore. So if we add it to our sham, we have some species that are able to absorb but does not emit fluorescence. But at this point, we don't know if they does not emit fluorescence at all, or just if their fluorescence quantum yield is lower, for example. We don't know if they does not emit or if they emit less than at the beginning. And beam is not enough to know that. So we performed also some fluorescence decay measurements. So here we photobleached at different irradiation time. And we see that the fluorescence lifetime always decreases during photobleaching. And if we look closer to the decay, we have always the same signature with a decay which at the beginning is really mono-exponential and then become bi-exponential, with one lifetime which is the lifetime of the starting protein and another which is shorter. So this means that here we have a new photoproduct, a new family of photoproducts which have a lower lifetime. And this one is particularly vicious because when we do fluorescence imaging, a lot of people use fluorescence lifetime measurements, which is called FLIM, and then the lifetime is the signature of your protein, and you use it as a probe of different biological events. So if it changes when you put light on it too much, it's a true problem. So then we have the colored, the dark species, and we have the damaged species, which is the one that I just described. And if we put all these together, we see that we have to add the dim species. So we have both absorbing species that fluoresce less, as I described, but also some that does not emit at all. This one I can't show you because it's just by the calcul that I can know that. So I can quantify all the contributions of these different families of photoproducts for each protein. And what we know is very interesting because we see that the pattern can be really different from a protein to another. If I begin by the most easier to describe, I have the YFPs that are really honest, I could say, because we are not bleached, or we are rarely bleached, in the sense that we lose absorption, or we are great. For the green one, it's nearly the same, because we have just the difference here with the dim species. But when we do fluorescence imaging, we just look at the fluorescence. So for this one, it's an on-off mechanism. You have fluorescence signal, or you have no fluorescence signal. It's binary. And we have the nice one here, because for M. cherry and the CFPs, we see that we are forming a lot of damaged species with the lower lifetime. And it's particularly problematic for this one, because we often use it at fret donor in fret film experiments. So we use the decrease of the lifetime as a marker of protein interactions. So this means that here, if we just bleach your sample without taking care about it, you can think that you have fret, even if you just bleach maybe your sample. So to conclude, we described here 4 spectroscopic signatures or 4 families of photoproducts for a wide range of FPs. And we saw that the different contribution of each photoproduct is really different between the different FPs. And we saw that one of these families is particularly problematic because it can lead to misinterpretations of film experiments. And also, a part that I can't describe it because of the time, we perform some mass spectrometry and SDS page experiments to see what's happened at the molecular level. And we saw also a wide diversity of photoproducts in a chemical way, because we have a lot of oxidation, but also dimerization and cleavage, which show that this phenomenon is clearly complex. So I want to thank the French Society of Photobiology, which found my travel to go here. Doctoral School of Paris-Saclay University in chemistry, which found my PhD, and the laboratory and all the people who worked with me. So thank you for your attention.